Good day, everyone. On behalf of BioIT World's Global Web Symposia Series and our sponsor, Genestack, I'd like to welcome you to Technology and Strategy for Building Multi-omics Visualization Tools that can rapidly adapt to user needs. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now I'd like to introduce our presenter for today. He is Kevin Dialdestoro, Master of Philosophy, Head of Data Science Consulting with Genestack. Welcome, Kevin. The presenter ball is yours.
So, hi everyone. Thank you for joining the webinar. We'll be sharing technology and strategy for building omics visualization. This is an important topic because many scientists are struggling to explore omics data effectively. Before we start, let me introduce myself and Genestack. My name is Kevin, and I'm leading the data science consulting team at Genestack. I've been doing bioinformatics for almost a decade now. I started my journey in Oxford, where I was inspired by the growing community of statistical geneticists like Peter Donnelly and Jill McVean, who made significant contributions to the 1000 Genomes and HapMap projects. My first bioinformatics project was to look at HIV evolution, and what made that project exciting and led to a publication was not so much in the algorithm that I developed, but in the high-quality HIV data that had been collected over several years. So that left an impression on me that data is the most important thing, not the algorithm or code to analyze it. It was true back then, and it remains true today. So, I'm excited by data, and we've gone a long way with multi-omics data, with single-cell data, and with population-scale data. So, what's keeping me and the Genestack team up at night? It's enabling people better access to high-quality data. Genestack was founded by Misha Kaposzewski, who was previously a group leader at the European Bioinformatics Institute, and he led the development of Expression Atlas. We've worked on various data transformation initiatives over the years in the context of drug development, clinical diagnostics, and agriscience. Some of you may have heard about our press release with AstraZeneca last week. We are building capabilities to organize and search their fast-growing multi-omics data across sites and across therapeutic areas. We are headquartered in Cambridge, UK, and we'll be opening a new office in Boston this year. So, everybody's been talking about data democratization. 9 out of 10 scientists are unable to explore omics data effectively in a typical R&D organization. This is what our clients complain about, and I've seen this firsthand when I'm giving training or consulting sessions. So, what could be the reasons? The main reason is that they are not computationally savvy enough, so it's hard for them to deal with the data directly and independently. They have to turn to data scientists or self-service visualization tools. But data scientists' time is limited, and bespoke self-service tools are either too hard to build or too expensive to buy. So, to address this, we want to make visualization applications easier to build so they'll become available faster for the data scientists. We expect this to have a big impact in a data-driven organization. Consider a typical scenario in a medium R&D department where you have, let's say, 20 data scientists, and 200 biologists. A data scientist would typically spend 10 hours per week for building visualization applications, another 10 hours supporting biologists for common tasks, and a biologist would spend 10 hours for exploring data. If we can make self-service applications faster to build and more available, a conservative estimate would lead to saving each of these activities by 50%. This translates to annual cost savings of at least $5 million. And not to mention the long-term exponential gain from better science. So, this is where we want to get to. Data has grown rapidly over the years. It's not uncommon now to want to query millions of cells across studies. And we don't want to wait for a year and invest $1 million for a single cell expression atlas. So the challenge that we've set up is, can we build these kind of applications in a day by a single data scientist? Can we outpace the growth of data and data types? The answer is yes, you could. What you are seeing here is an R-Shiny application. I wrote this application in a day with less than 300 lines of code, all in R, no Java, no JavaScript, no HTML, no CSS. Scientists can enter a gene or several genes and see in which cell types they are expressed. The cell types are mapped to a cell ontology, and this works not only for a single study, but across studies. But there's no magic. This is only possible if you use the right lever. Because the hardest part about building omics visualization is not in the graphical user interface or the graphics, but it's in organizing complex data and building a search capability that scales. So the lever that you need is solid data management and search capability. To take this point further, let's look at the common limitations with omics visualization apps. You'll find that the problems are not so much with the graphical user interface, but with the underlying data management and search capability. When you want to know where a gene is expressed across studies, the challenge is with scalability or with collecting and harmonizing different data sets. When you want to stratify individuals by their omics profiles, the challenge is in tracking lots of omics, multi-omics relationships. And when you want to correlate molecular and phenotypic data, the challenge is that they are often stored in separate files or databases. So it's hard to integratively query them. So your best bet is to focus on the underlying source of limitations, data management and search. After investing into data and search, you'll have the lever to build applications much faster. It's difficult to manage data and metadata because you need to break down data silos, clean messy metadata, and track complex relationships. It's also difficult to build search capability. As you get to thousands of samples and millions of variants, you'll be dealing with billions of data points. And you'll find that you need specialized databases or search strategies to query them effectively. Once you have the data and search in place, you'll find that building the graphical user interface a lot easier. And you don't want to invest in big, complex visual applications because maintaining them and extending them will be expensive. This leads us to what we call a data leverage strategy. What does this mean? It means you should invest into data and search first, and the applications will come easily. So easy that they can become disposable. You want a centralized multi-omics data management. So the data can be in good shape as soon as possible. Because organizing and integrating your data only at the point when you need it will be painstakingly difficult. Secondly, yes, building a search capability is difficult, but it can be highly reusable if you do it right. We recommend following the principles of service-oriented architecture. So you want to have loose coupling between services and have standardized ways of accessing them, etc. Third, when it comes to building the interface, consider lighter implementation in R or Python rather than Java or JavaScript. This will make the life of bioinformaticians and data scientists a lot simpler. They can integrate bioinformatics and data science packages more easily. And nowadays, you can build interactivity rapidly in RShiny or Python Dash. Again, you don't want to build a big all-in-one application. They tend to be complex and hard to maintain. You want to follow the Unix philosophy of building small, modular programs that do one thing and do it well. So this is the data leverage strategy, but we don't want to leave you with just a strategy because the execution is the hardest part. So we'll show you some of the technologies we have to make visual applications easier, cheaper, and lighter to build. The technologies are part of our product called Omics Data Manager, abbreviated as ODM. ODM will help aggregate and organize your data. What sets ODM apart is the ability to manage multiple omics data types, not just one. So you don't need separate data stores for different data types. You can internalize public data sources, combining them with your private data, and it will make them searchable. So rather than having to go through files and folders, you can quickly scan what data is available through facets and metadata summary, just like the way you would browse books in Amazon. Your visualization also needs to know the data relationships. Omics data can be complex, so we've designed ODM to be aware of different entities and capture their relationships. So, for example, here, you can track what samples are associated with what studies. And for each sample, you can track what multi-omics data are associated with it, along with the respective metadata, like which genome version, pipeline, or normalization method was used. And your visualization also needs good metadata, so users can understand the context and find relevant information. Using ODM, you can set metadata standards in your organization. For each data type, you can define what attributes should be captured and what ontologies or vocabularies should be used. You can validate and clean up thousands of samples quickly. And most importantly, you can minimize costly retrospective curation by getting the data right from the start via template-driven study design. Once the data is in good shape, your visualization needs to be able to query them. Using ODM, you can query across omics and across studies all at once. It's integrative, so you can query both the data and metadata at the same time. You can query individual metadata and data points like the tissues, cells, genotypes, expression values, and so on. Now, I will show you example API calls and Archani applications. The first example here is based on a subset of the TCGA dataset. What we want is to produce this survival plot at the bottom right corner. It shows that individuals who smoke and have KRAS mutation, overexpression, and high copy number alterations will have a much poorer prognosis. To do this, we only have to make a few API calls. First, the sample information by calling the search samples method. And then we want to get the variance and the expression data for KRAS. So I'm going to do this live. Wish me luck. So I'm going to make the API calls from an Archani, from an R Jupyter notebook. You can also do this from Python or via direct HTTP calls. So here, first, I specify the instance and my token. Okay. And next, I specify the study that I want to query. So I want to query this TCGA lung adenocarcinoma dataset. And I only want to get the male samples. So it's a pretty simple query. And in practice, you can do more complicated filtering. And this is the API method that we are using. After calling this, we immediately get the male samples. And here you can see their smoking status, gender, race, survival information, and so on. Okay, so we have 242 samples, so we can retrieve it very quickly. And once we have the sample information, you can then immediately compare the survival days between the smokers and non-smokers. Yeah, so this is just a regular R function to get the samples and to group them by the smoking status, and we get the average of the survival days. Okay, and next we want to get the tumor KRAS mutations. This time we call the search variant method, specifying the same study, so TCGA lung adenocarcinoma, specifying the same kind of samples we want to retrieve, and this time we want to get the genotypes for this gene. So we'll retrieve all the variants at this gene location. And again, in practice, you can filter more complicated can supply more complicated filters here by genomic regions and or annotations. So after calling this API call, here you see that we are getting about 2,500 genotypes from the 240 samples. And again, we can play around with this data set quickly and compare the survival by the KRAS mutation status. Yeah, so you can see that those who have KRAS mutation have lower mean survival days overall. Okay, and yeah, so it's multi-omics, so you can do this again for other data types. So next we want to get the expression data. This time we use the search expression method, and here we specify that we want to get the expression in FPKM for KRAS. Okay, so here again you get the expression values. You can get the expressions for KRAS for the different individuals. And again, you can look at the comparing the survival days average between those individuals that have overexpression for KRAS versus those who don't. Again, you can repeat this for other data types. So this time it's the copy number alterations. Okay, so here you can quickly see that individuals with high copy number alterations for KRAS have lower mean survival days overall. And finally, this is just a simple R code that combine this together, grouping the patients who are smoker and have KRAS mutation and overexpression and high copy number alterations. Okay. And as you can see here, there is a clear split in the survival probability. So in just a few API calls, you can create this complex analysis and visualization. And you can use these as building blocks for your interactive visual application. Which brings us to the next demo of an R-Shani expression atlas that's driven by these APIs. This application lets users to enter genes, filter by cell types, select one or several studies, and compare the gene expression across cell types and studies. And it's really easy to put this together. There are only 3 API calls that drive the search, which you can see on the right-hand side of this slide. The first call is to list and filter the studies. The second is to get the samples in the selected studies. And the third call is to get the expression values from the studies, the samples, and the genes that the user specifies. So you can query multiple genes and multiple studies at once, not one by one. And it's scalable. You can extend this for a single cell too. So this is a screenshot of the extension of the previous expression atlas for single cell. You can see the t-SNE plots here and the expression of some genes overlaid on top of the clusters. And we are continuously improving the performance. So let me show you how the Arshani application looks. One second. Yep. So this is a simple Arshani application using the APIs. You can enter some genes here. So let's look for CD3E, let's say. Okay. And let's say we are only interested in the lymphocyte cells. Okay. And we can select an RNA-seq study. Let's pick the first one. This is just an example of some studies we have in the in the demo instance. So here we can see that this gene is T cell specific, and here's the underlying API call. So this is the API call to find the studies under immunology, to get the samples from one of the studies that we've selected, and another API call to get the expression data from the study that we've selected and from the gene that we have specified. Okay, and let's look for an additional study. So let's take the second study. And here you can see the API, it queries multiple studies at once. Okay, in this example, it just supplies a list of different study accessions, but you can also apply a more general filter. Let's say you can apply this filter directly, query across all studies that have been tagged by immunology as the therapeutic area. And we can look for an additional gene, let's say CD28. Okay, so here you can see that we are querying two genes at once. So again, the idea is that you can query across studies and multiple genes or samples at once. And the plot will show the expression values for both genes across studies and cell types. Okay, and we can use a similar mechanism for single cell data. So let's clear the filters here. We are going to pick some single cell data as example. Okay, let's select one of the studies here. Okay, so we can see here under the Disney plot tab the Disney plot. So what you see here is about 50,000 cells across about 10 different cell types. And it's pretty quick, as you can see. And going to the sample tab, we can see the summary of the samples and cell types. And you can filter this down pretty flexibly. So let's say we are only interested in the lung squamous cell carcinoma. You can type in any filter here. So let's say we filter the samples by disease equals lung squamous cell carcinoma, and we have immediately fewer samples. Okay, and so the filtering is pretty flexible, and you can apply more complex filtering like using logical condition here, and tissue equals lung. Of course, in this example, it's not going to make any difference because all the samples here come from the lung. But that's the idea. You can apply complicated filters easily. And okay, and if we go back to the Disney plot, yeah, you can see fewer cells now because we have filtered down the samples to lung squamous cell carcinoma. And like before, you can enter a gene here and see which cluster has higher expression for this gene. So let's say, okay. So we are interested in which cluster this gene is more highly expressed. And you can see here that this gene is highly expressed in this cluster, which corresponds to the B cell, which makes sense because this is a B cell specific gene. Okay, and again, it's pretty flexible, so you can search across multiple studies at once. So in this example, we have 2 studies, and we can select both of them. And at the API level, we'll be querying multiple studies at once. Okay, so this is querying multiple studies for this gene, and in the Disney plot, you'll see the clusters for both studies. This is for the first study, and this is for the second study, and you can see also where this gene is expressed across both studies. Okay, so that's the end of the demo. Let me go back to my presentation. 1 second. All right, great. So we are coming towards the end of the webinar now. In summary, ODM can empower your data science and visualization applications, be it Arshani, Jupyter Notebook, or business intelligence tools. It does this by aggregating, integrating, and standardizing your data. It helps you to query billions of individual data and metadata points across studies and across omics. And by focusing on data and search first, you'll have the leverage for applications to come easily. So thank you all for listening. And thank you for my colleagues in Cambridge and St. Petersburg. The best part of my journey has been working with you guys. So thank you.
Thank you very much, Kevin. We do have quite a few questions that have come in, so I will get started. Our first question: in the current pandemic, working collaboratively can be challenging. Have you got any comments on how data can be worked on together and shared, or how visualizations can be produced collaboratively?
Okay, thank you, Elizabeth. So yeah, there are 2 parts to this question. One is how data can be worked on together. So yeah, as you saw earlier, we are pretty much our mission is really about making data more accessible. So as you've seen, we have a product that allows data to be aggregated from different sources, can share data to different groups easily. And what you'll have is a single portal for accessing all data that you have access to, from public sources, from private sources, for different data types. And it's also possible to write back your analysis results into the platform so other people can access them easily. So I'm referring to this, to this part of my presentation. Yeah, so you can see here all data organized in one place. So you don't have to go through different folders or files. It really provides a better experience. And the second part of the question is about how visualizations can be produced collaboratively. So that's a good question. Yeah, it's important to understand the use cases before building any visualization application. So we recommend close collaboration between the UX or UI designer with the biologists, the end users, and also the people who will be building this application. And our goal is to make the process of building this application much faster and much more natural. That's why we recommend, building the applications maybe in R or Python, so it minimizes the barrier for data scientists or bioinformaticians who have the domain expertise to write these applications.
Excellent. Thank you. We have a series of 3 questions regarding the data sets from Mike. The first question, how do geo data sets get in there?
Yeah, that's a good question. Thank you, Mike. So we have a way to programmatically connect to different public databases and pull their metadata and data programmatically. So Geo, in the case of Geo, yes, we have a connector that connects to Geo and pulls the metadata and make them available in our system. So you don't have to import them one by one. It's done programmatically, and we regularly check for updates and collect them. And once the data is in the system, you can harmonize the public data sets with your internal data sets using a common metadata template, ontologies, and controlled vocabularies. So it has a way to internalize public data sets like Geo, which is what I'm showing on the slide here.
All right, perfect. The second of that 3-question series, for internal data sets, who's responsible for entering the metadata?
Oh, that's a good question. Well, for internal data sets, you'll have to enter the metadata and data because it's your data, right? We do provide services to load data sets for you if needed. We also work with data curation companies if you need help curating your data. However, we do provide curation tools and study design interface so to help you enter your metadata more easily and to curate them more easily.
Thank you. And then the third question of that series, what ontologies are you using? Are they built in or can they be customized?
They can be customized. That's a good question. Yeah. So so the idea is that you can. Let me exit this screen so I can use my pointer. So yeah, the idea is that you can define your own metadata template and you can upload your own ontologies or controlled vocabularies to drive the validation and the search for specific metadata attributes here. So everything is customizable.
Thank you. Our next question, new data types become popular in the industry every few years. For example, single-cell RNA-seq. How would you keep up with managing and developing search capabilities for these new data types so that new visualization apps can be quickly built on this foundation?
Mm-hmm. Yeah, so we understand that the industry is moving rapidly. That's why we recommend investing into a good data management system that is flexible. And so we've built our data management system based on a service-oriented architecture. So it has a flexible architecture that it can accommodate new data types like single cells and other omics data types like ATAC-seq, ChIP-seq, and so on. So yeah, we want a system that can rapidly evolve. And in addition, it's also important to be able to capture diverse metadata. So we want our metadata model to be flexible and it should be able to capture new attributes that are specific to different data types. Lastly, we do have domain experts who understand the significance and use cases of new data types, and we can advise biologists on what apps they would need most. And it's good that we collaborate with different clients in the industry. So we like to learn new things, and we like to share what we've learned. So yeah, I think it's going back to the first question about collaboration. I think collaboration is important if we want to adapt rapidly. So rather than reinventing the wheel, we want to collaborate and try to work together.
All right. A question from Greg. You mentioned search capability that scales is one of the hardest parts in omics visualization. What enables your solution to scale, and can you give any examples of the kind of scale it can cope with?
Mm-hmm. Yeah, that's a good question. There are different ways of answering this. So so what we've done is that we don't limit ourselves to one search technology. So we try to integrate different search technologies that are optimized for different kinds of queries. Because one search technology may be good for one data type, but may not be so good for another data type. For example, a knowledge graph may be good for dealing with, let's say, metadata, where you want to model the semantics between different entities. But it may not be so good when you're dealing with structured high-dimensional data like gene expression data or variant or like a big VCF. Yeah. So what we've done is, yeah, we are using different technologies that are optimized for different data types. And it also depends on the kinds of questions you are asking to the data. One particular search technology may be optimized for one particular question, but not for other questions. So yeah, we try to be flexible with the choice of technologies for different data types and different use cases. And we use a combination of proprietary technology that we've developed ourselves, as well as public technologies.
Excellent. We have a couple questions for you from AC. The first one, can you curate and enter metadata via API?
Can you enter and curate metadata via APIs? Yes, you can. So yeah, you can basically do everything via APIs. So you can see here in this slide that in general, for any endpoint in the system, you can push data programmatically, you can edit them programmatically, you can delete them programmatically, and so on. So yeah, everything can be done via APIs and be automated. And yeah, there are multiple ways to enter data into the system using a combination of entering something via the graphical user interface and also entering something via APIs. So you can have a flexible workflow.
All right. And AC's second question, how is data versioned and provenance captured for any analysis? For example, in notebooks.
Yeah, so that's a good question. So what we've done is, so there are two kinds of versioning. One is metadata versioning, and the other is data versioning. So we try to do both. So we've supported metadata versioning in one version of our product, and we are going to add data versioning as well for soon. But the idea is that when you make the API calls, you can specify what version of data or metadata you want to use. So it makes sure that your downstream analysis is reproducible.
Thank you. Another attendee, Robin, asks, what are the system requirements for ODM?
System requirements for ODM. That's a good question. So we can provide this via software as a service. So you don't need to install anything. You can just use our cloud instance, public instance. If you want to have your own instance on premise, then we can deploy this via a set of Docker containers, and the system requirements are pretty standard. It depends on the amount of data that you have in mind, but yeah, let's say about it's pretty efficient. I would say. So I think a powerful instance with, let's say, 128 gigabytes of memory should be sufficient.
Thank you. We've had a question come in from Joe. Can you briefly give an example of how Genestack works with an organization already with large, unordered, multi-omics data sets?
Large unordered multi-omics data set. Yes, that is a tough position to be in. That's why we always preach to people, get your data right at the point of data generation. You don't want to clean them up later. If you are in that position already, we can help you. We provide services to clean up and to integrate your data sets. But there's no magic solution. I mean, we do have tools to do large scale curation quicker, automatically via automated script or also via the graphical user interface here. But it will still take quite a lot of effort to clean up enterprise scale data. So our advice is to avoid to be in that situation by making sure that your data is good right from the start.
Perfect. Thank you. A question from Robert. Does Dr. Dialdestoro believe that MATLAB may be used as a viable alternative to R or Python in managing some of these multi-omics studies?
Yeah, I guess it's not so much about managing, right? You want to use MATLAB for analysis, right? Yes, you could. The only consideration here is that the shareability of your analysis. Most people who are familiar with omics data are bioinformaticians who are familiar with R or Python, but not so much with MATLAB. So if you share your MATLAB work with your colleagues, you have to beware whether they will be able to understand it and manage it. Another consideration is with the availability of bioinformatics packages or data science packages. It's a lot easier to use them in R or Python rather than MATLAB.
All right, and we do have time for just one more question. Can you give us more detail on how the idea of lightweight Viz apps might work in practice to answer new scientific questions as and when they arise?
Okay, the question is how lightweight visualization apps might work in practice. To answer the new scientific questions quickly, yeah? Okay, so our Shiny apps, they are good as long as they are cheap and available on demand. So if a scientist says, I want this visualization, an organization must be able to deliver it within a week or two maximum. So it should not be a big development effort. Otherwise, scientists might end up spending their time on this. And centralized multi-omics data management solution and a well-documented set of APIs are important. Your organizations also need to make sure that you have the right expertise to deliver these applications. That's why we recommend R or Python based applications, because typically bioinformaticians or data scientists are the ones who build these applications, right? And they are mostly familiar with R or Python rather than Java or JavaScript. Yeah, that's it. And yeah, of course, we also can help you develop some of these applications quicker, because we have experience in developing them time and time again.
All right, thank you so much. We do have quite a few more questions. I will be compiling those and sending them along to our presenter for today, and you'll receive your answer via email. So I would like to very much thank our presenter for today, Kevin Dialdestoro. I'd like to thank our sponsor, Genestack, for sponsoring today's web symposium. Finally, I'd like to thank those of you who came and spent this time with us. We're very grateful you chose to be with us, and we hope we were able to provide some information that will make life a bit easier for you. So on behalf of BioIT World's Global Web Symposia Series, thank you all so very much. Stay safe and have a great rest of your day. Bye-bye.